Where's Leon? Leon, do we have we have slides ready to go? Almost? Oh. Oh no. You sure? Okay. Let me start. Okay, we are going to go ahead and get started with our afternoon panel, which is our time-honored favorite part of the afternoon of the Hope and Help Symposium. We're going to start with a short presentation from Synchron. So I'm going to introduce Nick Singer. He's the Senior Director of Strategy and Market Development at Synchron. Nick works with people living with neurological conditions, including ALS, and the clinicians who support them, to ensure that brain-computer interface technology is not only innovative, but accessible. He holds an MBA from Stanford and is also an alum of the Stanford Biodesign. Please welcome Nick Singer.
Thank you very much. I'm here today to talk about brain-computer interface and give a little bit of an overview about where Synchron is in our journey and some research that we've done to date and what's coming around the corner. Before I get started, I want to note this discussion is about a medical device that's currently under investigation by the FDA, and as a disclosure, I am an employee and a shareholder of Synchron. To get started, to kind of demystify BCI a little bit, just want to talk a little bit about mechanism of action. So individuals with paralysis, impairment, ALS, they still retain their intention to move, even if their limbs are not responding. So that area of the brain is still working and is still sending that intention to move. But that intention is disrupted. And we know that involves, that creates some challenges. Amanda and Ashley this morning talked about the ALS Focus Survey. This is data from that survey from 2020 that shows that the inability to do activities and limitations with mobility and walking were some of the top limitations and frustrations with ALS. And there are assistive technologies that are out there today that aim to make these easier, but they require that transmission of signal. The transmission of signal still needs to come to your eyes, to your voice, to your hands in order to operate them. And we know that there are limitations. We did a study of 150 OTs and speech-language pathologists working with your community, and only 6% felt that the existing technologies fully restored independence and autonomy. So there's more to go. And so that's where brain-computer interface comes in. What brain-computer interface does is you have an intention to move. That motor signal is transmitted, so the BCI takes that signal and sends it directly to a laptop or computer, bypassing the need to move eyes, voice, or hands. This effectively creates a brain-controlled mouse or keyboard that you can then plug into digital devices around you. The Synchron system that we are going to talk about a little bit today has 3 components. There's a stent, a transceiver, and a processor. What's a little novel about the Synchron system is that there's no craniotomy. There's no open brain surgery. The device is placed entirely in the vasculature. So just like stents that you think about for cardiac stents or stents for stenosis or for neurological conditions, it's set up and deployed entirely in the vasculature, and that's where it stays. There's no connection directly with brain matter. The transceiver is a device that sits in your pocket of your pec, kind of like a pacemaker or a deep brain stimulator. Once again, it's kind of a proven medical technology that's been around for a while. It's a very simple procedure. And then what happens is we have a processor that sits outside the body. It's there, you can see it on the shelf. What ends up happening is that takes all the data, and when an individual thinks, for example, of closing their hands, it turns that into a click on a Bluetooth device. And what we've shown in some clinical work is when you have these types of clicks that can be done with the device, you regain control and movement, or control over your digital activities and the digital world around you. So everything you see on the screen here that is moving was done with one of our study subjects. His name is Mark. He's up in Pittsburgh. And he's been able to do things like control his environment, his health care, work through tile boards and communication and shopping. Now, some of the things you see on this screen are being controlled by assistive technology that's actually on your iPads that you have today, but it's always the BCI that's providing the click or the BCI helping navigate through the screens. This is Mark when he was first getting his connection, and you can see it's a bit of an emotional moment for all of us. Maria is there, she's a Synchron employee, and the first time that we light up and know that we're gonna have that connection, and Mark's gonna have this throughout his progression, is something that keeps us going at Synchron, and something we hope to continue moving forward. So where are we in our journey? We've come a long way, but we have ways to go. So BCI as a whole has been 20, 30 years in development. Most of that work to date has been in the lab. What we at Synchron want to do is take this innovation that's been in university laboratory settings and bring it into the community. And so our first trial started in 2018 in Australia. Our founder is a physician at the University of Melbourne in Australia. That was with 4 subjects, 3 with ALS and 1 with PLS. Following out that study, there were no serious adverse events that were reported. Individuals were able to use the device independently at home. And this study was published in the Journal of American Medicine back in 2023. Synchron then moved to the States. Our offices are up in Brooklyn. And we did the COMMAND trial. And the COMMAND trial was a 12-month study. 5 of those participants had ALS. 1 of those participants had brainstem stroke. We reached the 12-month follow-up of that study last summer. For the primary safety endpoints were met, there were no serious neurological adverse events, and that study was recruiting out of New York City, Buffalo, and Pittsburgh. Our next clinical trial is around the corner. So our work has been finished for this clinical trial, and we're right now waiting for feedback and discussions to begin moving this soon, with the likelihood to start in the near future. That study will also be moving to sites that are outside of the Northeast to other parts of the country, and we hope to make sure that might include the state of Florida. This is still a feasibility trial. I need to really put that home, where we are early in our development process. So we look to once again demonstrate the safety of the device, and then measure the impact on autonomy, independence, privacy, and caregiver burden. And then beyond that, we hope to expand access. I lead marketing at a medical device firm, which doesn't mean that I get to come up and think of really cool advertising slogans. Marketing at med device means I get to understand the needs of those people that we design for. And in healthcare, that is patient first and always, but it's also caregiver, clinician, and implanter. And our ethos at Synchron is to design with our community and not for our community. We're very involved with multiple advocacy organizations within and outside of ALS. And my team has personally led over 250 voice of customer interviews with users with lived experience, clinicians, managing therapists. And one of the things that I'm here to tell you about today that I would implore you today, I don't have a clinical trial for you right now. But if you are able and interested in joining and learning more about this technology, is to join the Synchron BCI community. Right now, this community is a place to learn and shape. It's to learn about what BCI is, just the facts of where we are and where we aren't, to demystify the technology and explain a little bit about where we want to go in the future. But more importantly to me, very just personally for me, and selfishly maybe, is the shape. So this is where we go to make sure we're designing with the community and not for them. We go here when we say, which of these product features make the most sense? Which of these design screens give the most joy to the user? We go here when we say FDA asks us a question, and we can now quickly go out and say, oh, here's the response of individuals with lived experience. Here's the response of therapists that work with this disease, or here's the response of caregivers. And so if you have a moment today, I would really love for you to join synchronbci.com and join that community. Right now, as I said, it's a place to learn and shape. If and when we get to a chance to announce clinical trials, we will do it through here and also through the registry. Please join the CDC registry. That's a great place to go. Please do the focus survey. So I know I'm not up here to talk about the focus survey, but I just want to really implore you to do that. We used data from the focus survey in February in our FDA submission last month. So if ever you guys have a chance to ask for your voice, we here at Synchron, we care about it. We work with the association. We love the way to get your information in and make sure that regulators at FDA, at CMS and Medicare have it, because it's only with all of that that we can expand access to the therapy. And with that, that's all I've got for you today. Thank you.
Thank you so much. For the rest of the panel discussion, what I'd like to do is introduce our entire panel. Then we're going to hear 4 research updates talking about research that's going on at USF, Miami, Nova Southeastern, and Mayo. And then we'll open it up to your questions. Some of the questions were submitted online when you registered, so we have those compiled and sorted out by category. Some of you have filled out note cards during the course of the conference today. We'll get to as many of those as we can, and then we'll open it up to the floor as well for live questions. So I think we're going to save our Q&A for after all of the presentations have been completed. So let me start by asking, Dr. Ula, could I please ask you to start the introductions and just give your name, your clinic, and where it's located?
Can everybody hear me? Okay, hi, I'm Dr. Ula. I'm from Advent Health here in Orlando.
Hi, my name is Dr. Vahid Tuhiti. I'm the ALS director in Orlando Health.
Hello, my name is Nathan Carberry. I'm one of the neurologists at University of Miami.
My name is Jerica Farias. I'm a nurse practitioner and the associate director of the USF ALS Clinic in Tampa.
Good afternoon. I'm Eduardo Locatelli. I'm a neurologist at Nova Southeastern University ALS Center.
And I'm Bjorn Askerson. I am a neurologist and I run the ALS Center at Mayo Clinic in Florida.
Okay. And Jerica, you're first up.
All right, very good. All right, good afternoon. Thank you for your time and attention. As I said, my name is Jerica Farias. I'm a nurse practitioner at the University of South Florida. I first interviewed with USF in 2015, and as part of the interview process, my supervising physician, Dr. Vu, had me come to the ALS clinic and rotate through. And after the end of the day, he sat me down and he said, well, how do you feel about this? Emotionally, this is hard. This is a tough disease to treat. Do you think that you'll be able to handle this in 6 months? He was very blunt about it. And I told him that I would do my best to take care of the patient in front of me, to do the best that I could for them every day, and try to take comfort in the fact that I wasn't able to stop the disease by providing the very best care that I could instead. And I've spent the last 10 years of my career trying to live up to that every day. So right now at USF, we're in a little bit of a lull between clinical trials. We're hoping to have some things that we can offer to you before the end of the year. But what I'm going to be discussing to you today are some initiatives that have been led by nurse practitioners across the ALS community, across the country, that you probably wouldn't hear about otherwise. So firstly, I want to talk to you about the caregiver workshop that we began at USF. This project was initially kind of thought up of in 2019. I had worked for a professor who studied primarily caregiver outcomes in Alzheimer's disease. And what we found was that these caregivers had high rates of depression, anxiety, heart disease, sleep disturbance. And so it didn't take long working with the ALS community to recognize that our caregivers too are underserved. In 2019, my care coordinator, Kayla Martinez, and I drafted the first presentation that we wanted to develop. And of course, it being 2019, things really got derailed in 2020 when a pandemic broke out, because our goal was to bring caregivers in and give not just lectures, but hands-on demonstrations of equipment, transfers, things that we felt like, our caregivers are being thrown into this ALS world nearly as much as the patients, often underprepared, and there's really just so much time that we have in the clinic to share as much knowledge as we can, as quickly as we can, and so we wanted to dedicate a program for caregivers specifically. So this project was able to be restarted this year, and we were able to improve upon it. Instead of this being just Kayla and I, we were able to bring in members from the full multidisciplinary team. We had our first presentation in August of this year. We gave lectures kind of introducing patients to ALS and giving just a background education, kind of like what you had this morning. We talked about symptom manage, both psychological and physical symptoms that patients experience and ways to cope with them. We talked about national and community resources, dispelling some of the mystery about Medicare, Medicaid, long-term care, some of the misconceptions about eligibility that are out there. And then we also had our social worker lead patients through some different strategies to recognize their own caregiver stress and strategies to try to improve that. We did have a hands-on component of this. So Hoyer devices, speech therapists with letterboards and the speech generating devices, respiratory equipment, and we found the day to be really successful. We did do some pre-post surveys of satisfaction, and patients or caregivers were very satisfied with the presentations, and it is our goal to begin to offer them, twice per year, and we're looking at doing a remote attendance option as well. Another nurse practitioner-led educational tool that's out there is something called Essentials ALS. It's a website that was developed by Jordan Millard, who's a nurse practitioner at the Barrows Neurological Institute in Phoenix. There's a QR code here that you can scan with your phone, and it will take you directly to the website. But it's just a really nice, kind of simple tool. You or your caregiver would assist you to take the survey. It's basically modeled on the ALS FRS, which is a functional rating questionnaire that we use in clinic pretty often. And from there, it will develop a curriculum of patient education based on your current level of progression. There's a tremendous and overwhelming amount of information out there, and so this designs it to where you are in your journey and can be done, at a later time to give you updated information as your situation changes. One of the things that I'm really proud to share with you is some of the work that I've been doing with the Northeastern ALS Consortium. We all shared together that one of the most tricky symptoms to treat actually is thick secretions, so that tenacious phlegm and mucus that can get caught in the throat. We have patients who report, choking, staying up with it all night, and while we have a lot of pretty effective therapies for the excessive amount of thin, watery saliva, these thick secretions can be very challenging for you to experience, but also for us to help you with. And so we began work a couple of years ago, and this was led by Gabriela Lopez, who is a nurse practitioner at Nova Southeastern here in the state, but also with other team members through the NEALS APP committee. And so the first year we did a survey of 74 ALS clinicians, so neurologists, but also speech therapists, respiratory therapists, APPs like myself. And we got kind of the lay of the land, and it turns out that many people felt similar to us. Only 55% felt confident or very confident in managing thick secretions. We were all kind of using the same treatments, but there was a lot of concerns about treatment efficacy. And some of the challenges that we noted, beyond the fact that we didn't feel that all of the treatments were all that effective, was that, patients can have difficulty tolerating the treatments, or that, consistent use by patients can also be difficult. So cough assist can work really well for some patients, but if the hands are weak and they don't have a caregiver to assist them, it's not very useful. This year, we focused more on patient perspectives, and we did a survey of about 117 patients with ALS. 35% of them reported having thick secretions. We were actually interested to find that very few of them had existing lung disease, and only about half had a smoking history, most of it not, current. We found impact on quality of life in sleep, in stress, in social, ability to go out and socialize, in ventilator use. Similar to what we saw with clinicians, only about half of patients with ALS felt that the treatments were moderately or very effective. And despite having these symptoms and having this impact on quality of life, only about 3/4 of them were talking about it with their healthcare providers, which meant that 1/4 of them really weren't addressing the issue. And so that teaches us that we need to make sure that we're being proactive in, eliciting these symptoms from our patients, because it's very easy to focus on the things that are obvious, but the thick secretions can be something that is easily overlooked. So we have a QR code that will take you to the poster that we had on our 2024 results at the NEALS conference, and then we have an abstract presented to be, that will be given at NEALS this year. And the last thing that I wanted to close with was the MGH, or the Massachusetts General Hospital Quality Clinic. So to give you some background, Quality is the genetic therapy that's being used for SOD1 ALS. It's a complicated therapy to administer in that it's an intrathecal injection, or it's a treatment that's going directly into the spinal fluid. So it's not just something as simple as, you give it to a pharmacy and the patient takes it at home. It requires a lot of coordination, both in, insurance authorization, but time and, having the providers to actually administer the treatments. And because the current reimbursement for giving these treatments is low, that can sometimes limit how many places are willing and able to offer it to patients. I know that all of ALS clinicians want to give our patients everything that's available that's going to work for them, but the reality is that we work in healthcare systems that are not going to let us operate at a loss. So Jennifer Scalia, who is a nurse practitioner at MGH, she had the initiative to run a nurse practitioner-led quality clinic. And so they're overseen by a doctor, which I think is critically important. We absolutely need the oversight of our physician colleagues. However, the clinic itself is largely run by nurse practitioner staffs, which allows it to be a more cost-effective option, and they're currently seeing about 20 patients on a regular basis. And so it's, these types of models of care that may allow us to expand access to these important types of treatments. So hopefully next year we'll have more to report on in terms of research at USF, but I hope that you were able to learn some other projects that are available. All right, thank you.
Thank you, Dr. Carberry. You're next.
I think it's the bottom one. Hey everyone, my name is Nathan Carberry. Like I had mentioned earlier, I'm one of the neurologists at University of Miami. So my goal today is to kind of introduce you to some of the things that we're doing in terms of studies and trials at the University of Miami. I don't have any disclosures. The first thing I'm going to make a kind of a learning point and to remind ourselves the difference between a clinical study and a clinical trial before I start introducing you to all the different ones that we have going on at University of Miami. So, and of course, today you're going to hear about all the studies and trials, not just at University of Miami, but all over Florida. What is a clinical study? The intention of a clinical study is really to collect information to learn about a disease. So it's not really about intervention. It's not about necessarily a treatment. So it's collecting information, finding potentially biomarkers. This is really what leads to trials. Both are so important, and I'm making the distinction between the two because they're separately so valuable. And I think most people, when they think of research, they think, okay, so what's the treatment that I'm going to be taking? But studies lead to trials. So trial is really taking a specific experimental therapy and either receiving that investigational product or a placebo over a certain period of time and seeing if there's any response to it. And this, of course, is how we find out if a treatment works. But these two work together. So I'm really making a plug for participating not just in the trials, but also the studies. And you can enroll in one or both. So at University of Miami, we do biomarker, gene discovery, we do disease model development, clinical studies, and we do, of course, drug development, which is what everyone is most interested in. These things, again, all interplaying, working together. And the question I always get in situations like this, and the whole goal of this presentation is, how can you help? How can we work together to advance the therapies in ALS? The first one I'll mention, and this is a study, is CAPTURE. This is probably the lowest barrier to entry one that we have at University of Miami. Anyone with ALS or a related disease to ALS is eligible. The questions that we're asking are, what caused my disease? Am I getting the best management? And you can contribute just by giving us access to your medical records. And not necessarily any inpatient visits, any in-person visits are necessary. And the idea behind this trial this study is to discover patterns leading to disease. I mean, I very commonly when I make a new diagnosis, people say, well, I had this exposure, or I was in the war in Vietnam. Is it possible that this could be connected with ALS? We don't know. The only way that we're going to find out is if we capture all of this information and seeing if there's any reasonable patterns. Eventually, this could lead to new treatments, new trials. Remember, these things are connected. So trials is the next really big one that I'll mention. So this is clinical research in ALS. Anyone with ALS, a related disease, or even a control, so a normal control, would be eligible. The questions we're asking are, what tools can we use to detect disease? Because, of course, that's so important. We know many of us here, it took so long to get to the diagnosis. Sometimes over a year, the average is 12 to 16 months. So we need new tools. And the ones that we are currently using are MRI, nerve and muscle tests, cognitive tests, biological samples. Again, we're looking for patterns, looking for signals here of, is this connected with a disease? Is this potentially a target for treatment? We're hoping to find new biomarkers, and we're hoping that leads to new trials. This is a kind of a personal plug. It's a study that I'm working on with the University of Chicago, led by University of Chicago. Anyone with ALS, a related disease, or controls are eligible. And this is really targeting kind of half of the disease, which is not currently being captured with investigational tools. We all know very well here the dreaded needle test that really is only looking at the lower motor neuron dysfunction in ALS. But that's only half of the story. There's also everything going on between the brain and the spinal cord. So that's the whole idea behind this project. So this is really to hope test, validate, and investigate a new tool to capture upper motor neuron dysfunction, potentially even before it's clinically evident. And it uses only surface electrodes, so no pinches are needed. It's really painless, which is one of the perks. And if you participate, you're, of course, helping to validate a new electrical tool to make an earlier diagnosis. The plan being to help discover upper motor neuron dysfunction earlier and eventually to learn how to treat ALS faster. Because we always think that the earlier that we get to treatment, the better. This is really the biggest study that we have at the University of Miami. This is Michael Benatar's, his baby. So pre-FALS, pre-familial ALS. This is really what led to this field of pre-symptomatic ALS. And it's really amazing. It's been going on for over 15 years. And it's meant for people who have family members who are diagnosed with ALS. And then, unfortunately, they also were found to have a gene that potentially could lead to ALS. And then they're asking the question, well, what can I do now? What is there to do? So this is a natural history study where we are following people over a decade and just continuing to see how things evolve. The questions we're asking are, when does ALS happen? Is it like a starting gun? Is it like a dimmer switch? What does convergent disease look like? Is it, if we capture enough of these cases and document it clearly enough, maybe we'll learn something there. And then how do we prevent disease? I mean, that's really the golden question. If we know that we have a high risk for having disease because of a gene, is there something we can do to stop it from unfolding? So the goal here is really to understand that phase, all your whole life leading up to the point where you have ALS, if you do have a gene that's dramatically increasing the risk for getting this disease. And then, of course, we're always thinking about, can this help us treat patients better and treat people earlier? This is really the best way for family members to contribute. And like I said, it's been going on a long time, and it's not going anywhere. So I'm going to transition now into some trials, to medications, because, of course, that's really what we're all after, is finding the best drug and making sure it works. We've had so much excitement with this drug, and University of Miami really has played a big role in some of the investigations. So I just want to take a step back and talk about this medication, which was just mentioned about that Qalsody clinic. Qalsody is the same thing as tofersen. So this medication it's like a genetic therapy, and what it does is it prevents a gene, SOD1 specifically, from producing a toxic protein called SOD1. And it's administered in the spinal column, and it basically prevents a toxic accumulation of a protein that kills neurons and leads to ALS. So we have a recent study called VALOR that kind of led to its approval clinically. It was a phase 3 double-blind, randomized, placebo-controlled trial. The primary endpoint was not met, but like many trials in ALS, we wonder, are we just not looking long enough? Is the treatment period, we were talking about this in our group research meeting, clinical meeting earlier today. Because what we saw over time is that people that were on this medication had pretty dramatic response to the drug. These are 4 different endpoints that you can see a separation between the orange line, which is those that were exposed to the tofersen early, and the blue line were those that received the placebo. And it's, it kind of speaks for itself. So this is what led to the approval of this medication clinically. But before that all happened, before the FDA approved it, another trial called ATLAS was started. And this is the trial that's hoping to prevent ALS from happening, or to see if that's possible. And in that pre-FALS, pre-familial ALS, is really the study that laid for the groundwork for this to even be possible. So the questions we're asking in this trial are, is tofersen an effective therapy? We kind of already know it is at this point. But hopefully hoping to get even more substantial evidence with this trial. We're asking the question, does it matter when we start treatment? We have a kind of a strong feeling with any treatment in ALS that that's always important, but we wonder if it's going to be a dramatic difference with tofersen or Qalsody. And then we're asking, wow, can ALS be prevented? I mean, that's like a really powerful question. You get shivers even just considering that possibility. And so the way that the model for whether or not that can happen, that's all coming from the success of gathering biomarkers in those natural history studies. So again, another plug for doing both. Don't just go after the drug, do the study. So we're hoping to validate a new therapy, and we're hoping to substantiate the value of treating early. And my God, can we prevent ALS from happening? So the Qalsody Clinic is something that I started at the University of Miami. MGH is very hard to beat, but we're number 3 in the country. We are very, very high volume, and we're really, really proud of this. So anyone with an SOD1 mutation and ALS, of course, please come to me. I'll welcome you with open arms. The questions we're asking here, because we're enrolling everyone that comes to us into an observational study where we just see how people respond to the drug, and looking not just at the good things but also the bad things, because no drug is perfect, including Qalsody. And we're asking the question, how can I maintain a high quality of life with monthly infusions? Because that's a lot to do. And then we're asking the question, can I continue the treatment even though I've had a serious side effect? Because this medication works, but it does have toxicity and requires a lot of experience and kind of innovative thinking to manage some of the toxicities that come with this drug. So the hope with this observational study tied to an approved therapy is that we can benefit from and help understand a new treatment. And then you have the benefit of joining the 3rd largest Qalsody clinic in the country. You get to meet me, know me very well. And then you'll benefit also from our experience, not just from the clinical perspective, but just because we see so many of these patients from the research side as well. So we have a very unique amount of experience. And this is one of our patients. This is my founding patient for this clinic. She has made the news several times because she's had such a good response to the drug. This is her dancing for the first time since her diagnosis in the Keys. So really powerful stuff. And it's very, very proud of this initiative. It's very busy, but we'd be very happy to have you. Please spread the word. This is another really cool one. Sorry, you can take a picture later. So this is Neuralink. I'm sure you've heard it. This is on the topic of brain-computer interface. This is a bit different from the one that was just mentioned, the Synchron. This is more invasive. So this is for people that have ALS, but also spinal cord injury. So what we're asking here is, how do I communicate without the use of my voice? Can I control computers, devices, machines with my thoughts? And this is what the chip looks like. And this actually goes into the hand knob of your motor cortex. So the difference between this and Synchron, which I'm really learning about today, which sounds really exciting, is this actually implants directly into the motor cortex with many neurofilaments that receive information directly from the brain. And you control things by thinking about moving your hand. So it enables you to do things like play Mario Kart for the first time since getting a diagnosis. I mean, it's really, it's astounding. So I mean, this sort of field of research is really obviously going to take off. And University of Miami is one of 2 sites in the country that offers this. So if it's something that you're interested in, you can come up to me afterwards and I can get your information, and then we can see if you might qualify. We have other things going on. We're very, very busy at University of Miami. So we have some other trials, Astrals, which you'll hear about from some of our other folks on the stage. Healy is another one that's kind of in a pause, but we have other things coming. It's very busy, very exciting time, and of course, there's always a reason to hope in this field. So, thank you for your time.
Thank you, Dr. Carberry. Next, we'll hear from Dr. Locatelli and then Dr. Oscarson. And then just a reminder, after those last two presentations, we'll have time for Q&A on the 4 research presentations, and then we'll open it up to your more general questions, okay? Dr. Locatelli.
Thank you. Good afternoon. I work at the Kathy Hasman ALS Center in this beautiful building with an amazing team. Our goal really is to find the cure. We simplify what we go in the care. To find the cure, we need to collaborate with everybody, including the patients. We collaborate with other centers locally, nationally, internationally, and definitely we need to provide excellent care. So in our clinic, the goal is to, excellent care as we're trying to find the cure. And at the same time, in the university, we work with the students so we can, educate the providers that will take care later on. So I took a different aspect of how to show you all the research, and I took it from your side. And this slide is actually from Dr. Benatar in the University of Miami, and where they paint a little bit of a timeline of ALS. And you can see that at some point, the disease will have an onset, but before there are patients at risk, particularly those that have a gene positive, and then a family member has ALS, then they have a gene positive, and they are at a high risk of developing ALS. And there's a lot of research, particularly in the University of Miami and other parts of the world, looking to this population to understand when the condition starts so we can implement effective therapies really at the very beginning and sort of prevent ALS. And I'll show you some of the studies that are going on in that phase. So research dying in the pre-symptomatic phase. Now, once the patient gets symptoms, it will have some time until they get the diagnosis. But when it gets the diagnosis, we'll present to a clinic. And after the test and the diagnosis is sort of more of a set thing, whether it's definite or it's probable, then everybody will be offered to participate in some sort of a study or a clinical trial. Now, through this, I'll show you there's different kinds of studies. So in your minds, when you see, and I'll show you these studies, you can think, okay, where is my point in this journey? So you can see whether you may qualify for one or another study. So as you see, there's different stages. And here you will see that there's, before the diagnosis, usually you can involve these patients in what we call the natural history studies, but you can also be enrolled in this after you develop symptoms. So there's now one study called Prevent ALS, those high at risk, so they will be enrolled before they have any symptoms. But those that have already symptoms, they will participate in the second part of that study called Access ALS. And then I'll show you also there's other natural history study. Dr. Carberry just mentioned the very long study the University of Miami is taking, the PREFALS. But there's another non-for-profit organization running one called Target ALS. And they have a very ambitious plan because they not only include patients in the United States, but it's becoming to include also an international community to find diversity in the pool of genes and the pool of pathology they may have. So it's a very comprehensive study. So you see there's different entities doing similar things with different characteristics, but everybody sort of will have a chance to participate on them. Then when you get your diagnosis, early on in the condition, the person is, you guys can see, you're more functional. They can, is able to walk and talk and feed. Those patients usually will be asked to participate in what we call the double-blind randomized studies. You may have a chance to have placebo, or you may have the chance to be on medication. After a period of time, somewhere about 6 months, 9 months, or 12 months, depends on the design, everybody will move into receiving medication. It's called the open-label phase of that study. And sometimes you trying to participate in a study, you get to the screening visit, and your breathing is a little bit below the cutoff, so you're excluded from it. Or you find out you had a history of cancer, so you're excluded from it. or just past 2 years or 3 years from the disease onset. So they say, well, the study criteria is only for those at the beginning. So there's something called the early access programs, or EAPs. So in those studies, the medication is given to you, and then no placebo, and you're following as the double-blind study of the same drug is finished. So then, if we have an answer that works, then you stay on it. If it doesn't, if it's negative, then the study is completed, and then you can move to another study. And then in red, I put you can see some examples. Usually, when you get in the later stages, you want something to help you to move, and that's when you get into these BCI, so brain-computer interfaces like Neuralink or Synchron, and there's other studies of different methodologies that trying to interface the computer to the brain cells to trying to help you to move and communicate. So the natural history study, now we are involved into something called All-ALS, which is an NIH-sponsored study. Prevent ALS for those at high risk, and the ALS is for people with ALS, just symptoms got to be present. And like I mentioned, target ALS is very similar, however, includes an international community. The early phases study, we got involved with this now. The Healy study, which I'm going to talk later, is one that has phase 2 or 3 studies. This is called the MatchMat program, also developed by Mass General Hospital. There's now about 4 sites doing this. And it's to do studies trying to understand the dose of the medication, to make sure that medication gets to the brain, something called target engagement. For example, with the SOD1, means that that SOD1 protein is reduced, and also that the neurofilament is also reduced, means the medication is reaching target, it is doing the intention that it has, so they can, after the phase 1 or phase 1A study or 2A study, can move into the phase 3 study. So this is to accelerate all the pipeline compounds. We're trying to move them quickly into that. So people in the early stages of the condition, they can participate in this. We started recruiting for what is called the Acacia trial. This is a study on digoxin, which is a very old cardiac medication. Now has had a lot of background to understand that it can really reduce cell death and neuronal neuritic loss. So now it's a single arm. It's about 40 patients. 30 of them will be sporadic patients, and 10 patients will have a genetic condition called the C9orf. Completing these 40 patients will be preliminary to see if this meets this criteria of target engagement and biomarkers. If that's positive, then this compound will move into a phase 3 later on in the Healy platform trial. Here, the double-blind study of placebo controls, again, early in the condition. One group will receive medication, the other won't. The Healy platform trial is the largest. There's over 80 sites in the country. It's been running for a while. And they already went through 7 drugs, and 2 of them, CNMUA and pyridopidine, have moved into potentially doing phase 3 studies. But then the master protocols have changed. It used to be 3 years. Now it has moved to 2 years from disease onset. And also, the extended period, it used to be 24. It's going to be moved to 36 weeks. So there's some changes, and here's the update. This is one of the compounds that has been selected, a DP01. It's a transposon. Again, it's intended to help with the pathology. I don't want to spend too much on this. But essentially, there are some preliminary studies showing there is a good decrease in some of the biomarkers, and it's been very good in vital capacity. So this is a very hopeful medication that goes to the core pathology, the TDP-43 pathology and ALS. Another drug is called Monepantel, or Neurizon is the company. They has had also a study that shows very much that there's 2 levels of the dosing. One low dose was 39% in this low on the functional scale, and the other was 58%. So there's a lot of movement into this compound. Also, it's an oral agent. So these 2 compounds have been in there on the pipe, I think, by December, maybe January. The Healy trial will turn on. It's still enrolling patients. And I know that we are ready with all the paperwork, so we are waiting for the drug to arrive to our pharmacy. And as soon as that occurs, we'll be reaching out to start enrolling patients. There's potentially 3rd medication in there, which is also very interesting, but It's not being disclosed. I don't even know, but I know there's a potential third medication that will be enrolling in this trial. There's other phase 3 studies, the COYA 301, the masitinib, and Neuron. I'll specifically talk about the 301. This is a combination drug. It's a interleukin with abatacept. There's different mechanism of action, and they're trying to help these lymphocyte T cells to make them function better and suppress with the other medications some of the pro-inflammation that we can see going to the core of the condition. So this study is also around the corner to start. Masitinib has been around for some time. It's like 400 patients. They want to try to get some more recruitment. Has a very specific mechanism of action, but it still need to recruit. I think other sites are also trying to complete some of these patients to see if this study can finally end and give us some result and readout. And Neuron, this is kind of probably the most sexy of them all because it's a stem cell study. It's been around. They have had some trouble to confirm that it works. The company is trying to reopen enrollment and trying to see is a stem cell infusion, is patients' own stem cells. They're going to be obtained from the bone marrow. They're going to be sent to a special lab. They're going to be expanded first. Then they're going to be modified to produce particularly growth factors. And then those will be re-injected in the same patient every 2 months, but include also a placebo arm. So the patient may have, 3 infusions of, normal saline or water in the spine, and then other patients will receive the cells. But then after the first 6 months, everybody will receive their cells. So it's a double-blind study as well. It's a very aggressive protocol, and you have to be at the beginning of the journey to be able to physically afford to be participating in this study. The inclusion-exclusion criteria, I'll talk a little about that because everybody wants like this study or the other study, yet we have inclusion and exclusion criteria that you may or may not participate. And I'll tell you some of the AI components that we're trying to use globally to guide people how to choose a study. Talking about expanded access programs, if you already passed the 2 or the 3 year mark, or your breathing is a little bit below 60 or below 50%, depends on when your area, then you might be able to enroll in one of these expanded access programs. The first 2 here, the CNMU8 is, crystals of nanoparticles gold, has, a lot of energy there, but there are no open spots. So we have enrolled 45 patients in our site by now, but they are not given any more openings. There's a potential more opening funds from the NIH, so they might open new spots that yet to be announced, but nothing yet. The Pyridopamine also completed enrollment, so we're waiting to see if they open more, but those 2 we don't have enrollment. The Ibutolast, I think I'm going to leave it for Dr. Oscarson. He's going to talk about it. He's extremely committed to this program. He got a huge grant. He'll tell us all about Ibutolast. He's next to talk. And Spinogenics is one expanded access program. It's a compound that is bringing a lot of hope because it's supposed to regenerate neurites. The company seems to have a path now to be able to obtain this expanded access program. We think probably within the next month or 2 might be able to start enrolling. However, there has some caveats to this, but these are expanded access programs to offer those patients that don't qualify to enroll in the double-blind studies. These are the details about expanded access programs. And I want to spend a couple of minutes into this slide, which is very busy, but essentially these 10 points are the titles of the abstracts that our center is submitted to the NEALS, which is the National Consortium from Clinical Trials in the United States, and actually North America. We're doing a lot of research into Dr. Canberry called clinical studies. They are not essentially very, very interventional. There's not a compound, but they do give us a lot of information how to improve care, how to select patients. The first one is a retrospective time to event analysis that we've done in the center, actually 211 patients when we closed the database, and essentially is to try to understand the individual prognosis. You may have seen other folks that that got diagnosed with ALS, and some of them are going very slowly, and others are going very fast. So the progression and the decline is different among different patients. So for us to know who's going fast, average, or slow is very important. Not only to offer that, like, hey, worry a little bit, you're going fast, or like, relax, there's time to do things, but also to dictate what you're going to need in terms of your care. Also, understanding whether you may participate or not in a study, so on and so forth. So understanding your prognosis, I think, is key. So we look in detail in our population to trying to come with a model to understand the progression, the individual progression. Some of the things I can tell you, the most important is what we call the phenotype. There's different phenotypes that tell us there can be much slower or faster progression. And then something that we call the delta on the functional scale. So these questions, 12 questions that probably everybody has been asked here, the ALS FRS. If you do that the first visit and you get a score and you subtract that from the 48, which is the maximum, and you see how many points you're changing per month, that is your best predictor of how fast or slow you're going. Age also is a factor, your phenotype is a factor, and whether you got diagnosed very quickly is gonna goes against you. But some of those points are important for you to understand your progression rate. Then we have, Dr. Table Gray has done an amazing study here, studying the combination of lung volume recruitment. Essentially, they are the physical therapy for your lungs. So if you're at the beginning, we can give you lung exercises that are gonna improve your breathing capacity and maintain your function very well. So she's presenting those preliminary results. It's an amazing study. The third is a work we did in conjunction with the dental school. So we look at the teeth, the cavities, the bleeding that they had at the point of the probe to try to understand there was inflammation in people's mouth. So particularly bulbar patients had a lot of trouble brushing their teeth, and everybody else, with your hands may be weak, so on and so forth. So oral hygiene is essential, so we did a collaboration with them, and we learned very quickly that if there's no insurance in the dental to go twice a year to the dentist to have the formal cleaning, that is a problem. But now everybody receives recommendation to get an electric toothbrush and so for improve the oral hygiene. So that's a given now, and we're looking into expand this study for oral hygiene. There's another number of studies here, but our social worker, Lisbeth James, has done an amazing work here in association with the ALS Association, looking at other factors that determine your health, and we call social determinant of health, like your insurance, whether you have caregivers, whether you're far from a clinic, what is your income, and other factors that might impact on your progression. So I think that she has found that very useful, and now she's expanded into that research, and I'm very happy about it. So we have, you saw Dr. Charbet this morning talking about GI and tolerance. We're looking into those feedings and whether they can tolerate it if we give them before putting the peg. So these are practice improvements that we have to do as we go along to make sure that we, as we trying to find the cure, we improve our care as well. So we characterize the supplements. We have a nice presentation in number 9 today, which is the thick secretions in ALS. That is a great study because secretions are, really confusing when we put the ventilator. It's very uncomfortable. So we are learning about learning the 6 secretions and the combinations of how to manage them, and trying to stabilize, a standard of care across the clinics. And then also how we can improve the non-invasive ventilation. Actually, non-invasive ventilation is like a treatment. So when it started early on with the right settings, using non-invasive ventilation when it's not absolutely necessary is actually beneficial for as a disease-modifying treatment. So this is some of the things that we have put in addition to our clinical trials, whether they're natural history, whether double-blind, whether they're just phase 3 studies or expanded access programs. We also have a number of ongoing trials to improve our care and actually be the beginning of data to actually later on expand in other studies. I know nobody has PLS in this community. Maybe somebody has it there, but we also, as one of the related disorders, we are looking into PLS, trying to see if electrical stimulation can decrease the spasticity. If that's the case, this treatment might help also ALS patients that have that spasticity, which is cause such a discomfort and poor quality of life. And with that, I want to show you the team that we have. And as a Center of Excellence, we want to thank the ALS Association for all the support they give us and continue work and for making this amazing meeting today and give us the opportunity to share our research with you. Thank you very much.
Good day. I'm Bjorn Askersen. I introduced myself earlier, I think. I would like to thank the organizer for having me and all of y'all who are here and the other speakers who have covered a lot of the stuff that I had on here. I will echo some stuff and also talk about some specifics, some of the things that we are doing at Mayo Clinic. The big question remains, I think, at least in my mind and for many of us, is what is ALS? And today we call ALS based on a problem in the motor nerve system, but it doesn't truly reflect why these nerve cells are dying. And a lot of these studies we're doing is really trying to dig into that to see if we can understand why these nerve cells are lost. If we understand that well enough, maybe not perfectly, but well enough, then we can really do something about it. For most people with ALS, we think it's a problem with this protein called TDP-43. But for some people, it's a problem with this SOD1 protein. And you already heard about how successful we can be with treating that now. This is, the most hopeful time I've ever been in this field. And now that we are rolling out large clinics, I have patients of my own who are receiving this treatment, who are getting better. It's amazing. It's really, that was the dream when I went into this field. Now, of course, I wish I could give this for everybody. I had something that powerful for all, but we have it for some today. And it speaks to that if we understand enough of what is the problem, then we can fix it. So we're doing lots of projects. I'm sort of topping this list here with the project that we're doing. Dr. Petruccelli is one of our big neuroscientists. He's working with Dr. Benatar at University of Miami on this first project there, where we're trying to understand if we can measure that fundamental problem with the TDP in people, not just after death looking at it in brains, which we can do pretty good today. But we wanted to get to be able to really speak to if that molecular underpinning, the best molecular underpinning that we can see today of ALS, is there in people. That would be very helpful to get to an early diagnosis, to monitor drugs that we develop so we can kind of measure if a drug works or not. So that kind of test will be useful. Number 2 on that list there is another trial we're doing. We have more trials on the list there. Actually, we'll come back to this list and I'll see which other ones we kind of touch on there. And I can do that myself. So we've been doing this at Mayo for a bit. That is why there's so many projects that we have. Many of them are not active. Again, there's not that many that we're actually focusing on right now. But we do both these attempts of understanding what the disease is in its molecular nature. Oh, how did that happen? And then we try to apply that by helping develop products that can address it. We measure ALS in the people that we follow, and we work with people who have ALS to manage their symptoms as best as is possible. And again, research can be done at all these steps. Again, understanding what the problem is, trying to address it based off our best current understanding, and then how to optimize the treatments that we have. Some of the stuff that have been done with groups in Florida may already have touched you. So neurofilament proteins are something which you probably, most of you heard about. It's certainly been groups that Mayo been working on for a long time, groups at Miami has been working on it. Lots of groups throughout the world have been working on getting this test done. It's a test that we can now use. It can help us make an early ALS diagnosis. It's far from perfect, but it's a nice complement to the other tests that we have. it can also help us make guesses about how someone's disease will progress. That's something which is still difficult for us, even with this additional tool, but we are better at it with this. If we have a large group of people and we group them up and take an average, we can have pretty good ideas. For individuals, well, maybe not very robust yet. We're also looking at this in clinical trials more and more again. We probably didn't touch that much on how we need to set up these experiments when we do them in people, but often we have to be very selective, just picking people with certain characteristics, which leaves most people with ALS behind, which is very frustrating for everyone and makes trials much more expensive and difficult for us to do. And if we could include more people in trials, we would get the answers faster, they would be more representative, they'd be cheaper. Just lots of good things that would come if we can do it. So again, neurofilament right now is transforming how we do trials. And it was the main measure that was used to get that Qalsody drug that you've heard a couple of times about here approved. So we're already using it. We'll be seeing more of it. All right. Didn't I have other stuff on there? But yes, there's other things we're trying to do there. We're looking to kind of try to measure TDP and that our current the core of the problem is we can see it today. We do a lot of discovery of genetics, and again, our groups discovered the C9 gene, the most common genetic cause of ALS, back in 2011, and we're still hunting genes. There's lots of other groups in the state that are also hunting genes. Genes won't give us all the answers, we don't think. Again, environmental factors are likely somewhat more important than genetics. So we're still going to find more there, and those could be treatable things that we can get to quicker than we can get to the more not genetically defined ALS. But genetics won't give us all the answers. We know that, but if we can save some that way, we will take that. So it's definitely a hot topic. And it might inform us about ALS in general, too. We do a lot of tissue banking. That means that people donate their brains and spinal cords to the Mayo Clinic Brain Bank. That's after death. It is still, I would say, where we learn the most about the disease. We tried by creating animal models and stem cells from living people with ALS to study ALS. And there's lots of things we can learn that way too. But actually looking at the cells that have ALS often is the best way of understanding what's going on. We touched on that. Another tool that we have, and I think it's also coming, we have a very strong magnet camera, an MRI, where we can see most ALS now, where we see the part of ALS that's in the brain. The part of the ALS that's from the spinal cord out to the muscles, we don't see because, again, we're looking at the brain. But we can see that part of ALS that's in the brain, and it's pretty good. The regular strong cameras, now the 3 Tesla cameras that are more common, can also do this pretty good. So this is something that is rolling out. More people are recognizing that they can see it, and if you know what to look for, you will see it. Like any other diagnostic test, it's not perfect. It's not as good as that needle test, the EMG, but it's good. And again, it looks at the other part of ALS. Again, so as I said before, we currently define ALS by where, meaning both in the brain and out in the spinal cord going to the muscles. And this is a way that we can actually get a picture of that brain part. Genetic risks, well, I already touched about that. These Qalsody patients, again, you saw somebody dancing here. And again, I've seen that too, and it is wonderful. I wish it was for more, but we do have our first wins, and I take tremendous hope from this. And then we tried to collect all these samples to achieve that understanding. We have long-standing biomarker collection protocols where we collect things at Mayo. We also work with Target, ALS that you heard about, which is a large organization, has a lot of private funding for helping to collect biofluids. We work with All ALS, which is the NIH biofluid collection protocol. You, as a person with ALS or a healthy volunteer, can contribute to many of these. And when we use you in one of these studies, you get the unique identifier. So that when you get, so again, if I need 10 samples of people who are 40 to 50, male with ALS onset in the left hand, for some reason, that's kind of what I need for my experiment, and I request samples from these other organizations, we can see if we're getting samples that are actually from the same person, which could be possible if I have a very narrow inclusion that I kind of what I'm looking for. And these global identifiers make sure that we know if we're really seeing the same sample over and over again from several different studies. Because there's, again, the way we're doing this, we're trying to get more and more samples. And it is valuable. Even just because you gave to one study doesn't mean you can't give to 2 others. We keep track of it, and your gift may actually be even more valuable. Sometimes those, when we get these longitudinal samples, though they are more valuable. Most of the time, when we do longitudinal sample collections in the protocol, we really try to ensure that there's a certain time between them and so on. But occasionally a question comes up. I had a patient who had a thrombosis from a type of antisense oligonucleotide, one of these tofersen-like drugs, not tofersen, not Qalsody, but a different one. And we were trying to get samples from before and after that event. And we were able to do that by looking in different studies and pulling samples in. So there's a way to contribute more than once, please. Again, once is great if that's what you can do, but if you can do more, we'll take it. So this brain bank, I mentioned that. So there's still a lot of things that we don't know about ALS. Again, we know the nerves to the muscles get sick. That's kind of the basic premise. It's the cells from the brain to the spinal cord and the ones from the cord out to the muscle. If you only have problems in the top part there that we could see with the MRI, that's what we call PLS. And you heard that term used just recently here. And if it's both, we call it ALS. Now, in our brain bank, we have a very large series of people who have PLS. And one presentation paper that we have that's in review right now mentions a protein that was discovered a few years ago, ANXA11, being really a differentiator. So we can look at brains now and say if it's ALS or PLS based on how much ANXA11 is in there. And that's really interesting. Again, if that holds water, it would really change how we view ALS versus PLS as separate entities. There are other groups that also have seen this now, and we're chasing this to kind of see what this means. There are people who appear to have PLS and then turn into ALS. So how can they really be different than on a proteomic basis? There are still unanswered questions here, but this is definitely interesting. When we get findings like this, it certainly leads to thoughts. Could we limit the involvement by adding ANXA11 to regular ALS? Would we keep the disease only in the upper motor neuron region then? I mean, that would be not great, but still much better. Might be problematic with this particular protein because we also see that it also appears to go with an increased risk of dementia. So, but we're definitely chasing that one right now. So to get to the cure, we need help. We need more data. And again, you've heard about lots of different projects here that you can help out with. And we'll take the help where you can give it. I should come back to Butylast here since that was sort of said that I would. And I think that's true. It is a molecule that I'm very curious about. It's a drug that has been approved in Japan for the treatment of asthma for many years. It's a phosphodiesterase inhibitor. It's a gentle immune modulator. We know that the immune system is involved in ALS, and many of the products we're doing are targeting the immune system. The immune system is in many ways easier to get to than the nervous system, and It's easier to measure. So we're trying this drug Ibudilast both in a phase 2/3 trial that is fully enrolled, so we should have a readout in a little over a year. And we are doing an expanded access program where we are using this kind of neurofilament biomarker to determine if we can see a effect in some people. Our current definition of ALS, as I came back to, started with, isn't really what the problem in your nerve cells are. It's that you have problems in those nerve cells. So it's entirely possible. And we know, for example, for these SOD1 patients that we can treat with Qalsody, that it's a different reason than most people with ALS. So are there going to be people who respond to certain treatments and not to other treatments, who have this, what we call ALS, which is a pretty large umbrella today? It's possible. This way of approaching that question, both through a randomized controlled trial like we typically have, and in an individualized neurofilament-based expanded access program, are different ways of looking at it. So definitely trying to get to answers from different ways. Lots of people to thank. I have my nurse coordinator here somewhere. She's probably sitting in the other room. But there's tons of other people to thank, including the association and everybody else I work with here. Thank you.